All right, in this video, we're going to talk about regulation of gene expression again, but we're going to specifically focus on the operon, which is primarily a prokaryotic gene regulation mechanism. Apparently, they can be found in eukaryotes as well, but I've only ever known about the ones that happen in prokaryotic cells. And so we talked a little bit about regulatory sequences, and we said that regulatory sequences are essentially groups of genes that are regulated together. The AP Bio course descriptions calls this coordinately regulated, which seems a bit excessive. Coordinately regulated essentially means that they're all regulated together. They're all turned on together or turned off together. And so that's essentially what that means. It is a technical biology word, but it seems a bit excessive. And so basically what's going on is there's some sort of molecule that will trigger the expression of this or stop the expression of this operator sequence or operon sequence. And so operons, just a couple of more things about operons. Again, all the same regulatory sequence. They are characterized by this structure called an operator. This place here is going to be called the promoter, which is kind of like basically where the RNA polymerase is going to line up. Remember, this is the DNA here, and the RNA polymerase wants to come in and transcribe this gene in order to make proteins, right? And so what the operon is doing is controlling whether or not those proteins are going to be made. And so what we're going to talk about is one called the Lac Operon, which is commonly found in bacteria. And the Lac Operon essentially will make a series of genes that will be used to break down lactose. Lactose is a disaccharide that is composed of two different sugars. I know that glucose is one of them. I think the other one is maltose, just off the top of my head. And so lactose needs a special sort of enzyme to be broken down. We have the same trouble in our own bodies. Some people that don't have the right proteins to break down lactose are lactose intolerant. That doesn't mean we have an operon like this, but you get the idea. And so there's no need to be expressing these proteins unless there's lactose present, right? And so this repressor here is going to be bound to the operator as long as no lactose is present. There's no need for the cell to be making the proteins that break down something that's not there. You get the idea. But all of a sudden, when lactose, let's say bacteria comes in contact with lactose sugar, then that lactose will actually bind with this repressor. And you can see this is called allolactose. It's just basically the same thing for our purposes anyway. And so it binds to that repressor, which is going to then allow RNA polymerase to transcribe those genes then to begin breaking down lactose the way they normally would. And so before we talk about the other factors, just to make sure we understand the parts of an operon. If you see an operon on the exam, you're probably going to see the Lac Operon. You may see another one called the trip operon, which kind of works oppositely, but it's the same idea. All right. This molecule lactose is generically, it's called the inducer. So think about it. It's inducing, it's causing to happen this regulatory sequence. It's causing it to fire by binding with the repressor and causing that repressor to be lifted off of this sequence of genes and causing them to be transcribed, which is going to make the proteins that break down lactose. So what's going to happen then when all the lactose gets broken down, including this piece right here? Well, when all the lactose gets broken down, the repressor will bind again with the operator, and that protein will cease production because there's no need to make it anymore. There's no more lactose. So you get the idea. This is a really simple kind of regulatory sequence that is found in prokaryotes. There are other factors that can affect the production of this particular Lac Operon. And one of them is the presence of glucose. The amount of glucose can also help gene expression too. Think about it. If lactose is present, but there's already a lot of glucose, then there's no real reason to break down that there's no real reason to break down all that lactose because you already have plenty of glucose. And so that will say keep the operator off. But if there's no lactose and there is glucose, same thing, right? Basically, if there's lactose but there's no glucose in the blood, basically there needs to be an input of energies for the organism to stay alive. That is when the operon is going to be turned on. And so you kind of get the idea there. I mean, because remember, glucose is just part of lactose, and glucose is how cellular respiration occurs. And so as long as there's glucose available, the operon doesn't need to be running. But if there's no glucose available and there's lactose available, then turn on the Lac Operon and start the process going.